All right, great. Thanks for your patience. My name is Rebecca Albright, and I'll be speaking with you today about some of the challenges to coral reproduction and coral recruitment in a changing environment. So I am currently an assistant curator at the California Academy of Sciences. I started there last summer. I've been there about 9 months. I got my PhD at the University of Miami in 2011. And I was working with Chris Langdon and Margaret Miller and several other people, mostly on effects of ocean acidification on coral reproduction and recruitment. And after that, I was at the Australian Institute of Marine Science for several years and at the Carnegie Institution for Science at Stanford for a couple of years prior to starting at the California Academy of Sciences. So my most recent work has actually been on scaling and trying to understand ecosystem metabolism. So it's been a few years since I've done this type of experimental work trying to understand perturbation effects on reproduction and recruitment, but I'm going to do my best to try to give you the current status of knowledge in this area. So, okay, so it's of particular concern that what we know that reproduction in early life stages are critical for coping with environmental change, and that's part of the reason why we do what we do. But there's been increasing understanding that early life history stages in particular are very, very sensitive to perturbation. There have been a variety of different studies that have looked at this, and there's a lot of different perturbations out there. So ranging from global stressors that are increasingly on everyone's radar, like ocean warming and acidification, to local stressors that have been on everyone's radar for a very long time, like overfishing and water quality. So historically, there's been a lot of research surrounding water quality impacts on reproduction and recruitment. And then I would say over the last 10, 15 years, the effects of warming and acidification on these early life history stages has really kind of blossomed in the scientific literature. So these top two areas of warming and acidification, with a particular emphasis on acidification, because that's my area of expertise, that's what I'm going to focus on today. My talk is going to be focused largely on challenges, and then we have a talk from Madeline later this afternoon that is a bit more potentially solutions focused. So hang in there with hope. So just to contextualize some of this, this is from CO2earth.com last night. I actually pulled this off. So the current atmospheric CO2 levels are around 409 parts per million. And just to contextualize this, when I started my PhD in 2005, they were around 380 parts per million. And then every time I give this talk, every year, I kind of pull down the recent updates, and I've just seen it kind of tick, tick, tick up over the last 12 years. So when these slides are coming from a Nature paper that was released a few years ago, so in the lower left, this is increases in carbon dioxide that's in the atmosphere from the 1960s until now. And so this data are coming from an atmospheric station at the top of Mauna Loa volcano in Hawaii. And when this station was implemented, the atmospheric carbon dioxide levels were around 316 parts per million, and that was back in around 1960. So 316 parts per million, which is not that much higher than pre-industrial values of around 280 parts per million. So it's really been over the last 50, 60 years that we've seen these unprecedented increases in atmospheric carbon dioxide concentrations. And when this article was released, they saw atmospheric CO2 surpass 400 parts per million for the first time in about 3 million years. So it's a threshold that's particularly concerning. And when that carbon dioxide is released into the atmosphere, there's no real barrier between the atmosphere and the oceans cover the vast majority of the surface area of the planet. So just via gas exchange, about 25 to 30% of whatever's in the atmosphere just gets absorbed by the surface oceans of the globe. And that's doing us a service because it pulls that out of the atmosphere and actually mitigates some of the warming that would happen if it was kept in the atmosphere, but it's not without consequence. So there are chemical reactions that happen when CO2 reacts with seawater, and these are the cumulative effect of those is known as ocean acidification. So on the right, we see that with increasing atmospheric carbon dioxide, pH of the surface oceans decreases through time as well. Many of you are probably familiar with these concepts, but I just want to take a moment to not necessarily go through the chemical equations, but just introduce a couple of jargony terms that might slip out during this talk. So I just want to familiarize everyone with them. So when CO2 dissolves in seawater, it forms an acid called carbonic acid. It's a weak acid, so it dissociates rather quickly into its ionic forms. The net result of that, shown here on the bottom right, is that your carbon dioxide level is increasing, your pH is decreasing, your carbonate ion is decreasing, which is important for anything that secretes calcium carbonate, like many marine calcifiers. And then the saturation state of calcium carbonate also decreases. And what the saturation state is you can basically think of it as a proxy for it's the solubility. It's basically a proxy for solubility of a certain mineral phase. And so with calcium carbonate, there's various mineral phases. Corals secrete aragonite, which is there's high magnesium calcite, which crustose coralline algae often secretes. It's more soluble. But basically, if you have a saturation state that is greater than 1, it means it's super saturated and that things are going to precipitate more easily. If it's less than 1, it's undersaturated and things are thermodynamically are going to favor dissolution. So the reason this matters is that for marine calcifiers around 12 years ago, everyone started to understand that anything that secretes a calcium carbonate skeleton, either for protection, for support, like you and I secrete a skeleton, it makes it increasingly difficult for these organisms to secrete these skeletons. So they're expending much more energy, and there are energetic consequences of that in terms of a finite amount of energy that has to be allocated towards all your different physiological processes, and if you're putting more towards calcification, there's less left over for things like reproduction, which we'll talk about a little bit later. So when I started this work, most of the work that had been done was looking at calcification impacts on adult corals, and I became interested on what the impacts were on the next generation of corals on early life history stages. So I'll just walk you through a little bit of that today. Again, not hitting on all of the research that's out there, but hopefully trying to create a current state of the knowledge in terms of where we are with this. So we know that recruitment is determined by primarily by three sequential life history. There's larval availability, which is related to reproductive output, fecundity, and then fertilization success. And then there's settlement ecology. So once you have your larvae in the water column, they have to actually settle. And that is a complex interaction between the actual physiology of the and the quality. And then if they're able to settle, able to grow quickly enough to stake their claim on the reef and survive. So these 3 different phases are what we were looking at in terms of perturbation effects. Oh, and just noting that environmental factors, that because these are existing bottlenecks in the life history or the life cycle, that any environmental factor that disrupts these has the capacity to cause downstream effects that kind of accumulate through the life cycle and can have pretty devastating consequences at a population scale. So starting with larval availability. In terms of impacts on colony fecundity, and this is actually something that hasn't been as well studied as we would like because it's difficult. Gametogenesis in some coral species can last upwards of 9 months, so it's difficult to do these long-term perturbation studies. But there have been some papers published that indicate that prevailing environmental conditions influence reproductive processes through energetics. So again, this is going back to that concept that you have a finite amount of energy, and if you are expending more energy, either producing reactive oxygen species, if there's environmental stress going on that you have to try to mitigate, that's less energy left over for reproduction. So there have been a couple of studies that show that sublethal stress, in this case relating to temperature, like bleaching that you then recover from, can affect successive 1 to 2 gametogenic cycles and cause reproductive failure or reduce egg numbers and quality. And we've also seen environmental or ecological studies that show reduced fecundity with previous bleaching or in sites that are characterized by extreme conditions. These are large fluctuations in temperature. We should note that these are very species specific. Almost everything that we talk about in this talk is very species specific and genotype specific too. So different species respond differently, different genotypes respond differently. And it interacts also with local abundance, how many individuals are on the reef, and then whether or not they're able to successfully fertilize. So there is no one size fits all for anything that we're presenting today, which is one of the areas of hope, that there are resistant or more resilient species or genotypes. So impacts on fertilization and larval development. So this was one of the things that I did during my PhD was working with Margaret Miller and Chris Langdon, and we went out and we would collect gametes of a Acropora palmata and a variety of different species and then cross-fertilize them under different conditions. I was looking at CO2 and temperature effects. So this is an example of a cross-fertilization experiment that we would do, a few different sperm concentrations and different CO2 concentrations. And what we would do is collect the gametes, separate the egg from sperm, and then just cross-fertilize them under seawater that had been manipulated to be representative of a certain parts per million of carbon dioxide. These are what we score fertilization. So on the left are your no sperm controls. And what we were looking for is we wanted to see how the fertilization curve would shift under stress. And so your typical fertilization curve for a lot of marine invertebrates looks a little bit bell-shaped, where at low sperm concentration, you get low fertilization success, then you get increasing number of sperm and you reach an optimal fertilization, and you can actually get too many sperm. Results in something called egg will get fertilized multiple sperm, and then it actually makes that egg inviable. So there's kind of this sweet spot in the middle where you have an optimal sperm concentration. For a lot of coral species, that's around 10 to the 5th, 10 to the 6th sperm per mil. So this is why we did this studies at a variety of different concentrations to see what was happening. And so we're looking to see if that curve shifts, if it shrinks, if it morphs, kind of what happens overall. And what we saw, so these are results for Orbicella fabiolata, I'll show a few different species here, was that under elevated CO2, this fertilization curve was shifted to the right. So basically, in order to achieve the same fertilization success, you need more sperm. For this particular species, in these scenarios, we saw that you needed about 10 times more sperm in order to get the same fertilization success at elevated carbon dioxide levels. So we repeated these with a few different species. When we moved to Australia, we continued with Pacific corals. So this is a cropper. Time we added in warming. And what we saw here was one stressor, either carbon or temperature, you needed around 10 more sperm to get the same success. If you had both stressors, you needed about 100 times more sperm to get the same fertilization success. And so these stressors are acting synergistically, and so that kind of points to the importance of these multi-stressor experiments because there can be interactions, and it's not always negative. There are cases where warming can offset the effects of carbon dioxide, but again, it's very process dependent and sometimes even species dependent. So understanding the nuances of these is but the reason is that, first of all, these results kind of show that if you have a lot of sperm, these stressors don't seem to matter. If there's enough sperm swimming around, things get fertilized anyway. But when you're sperm limited at lower sperm concentrations, what we think is happening is that it's affecting sperm motility, and they can't swim as quickly, they can't find the eggs as well. And so that's when it comes into play. And in a lot of areas where we already have kind of deposited, or we have populations that are kind of low in density, you're going to be sperm limited. And then these are organisms that can't swim around and find a mate. So as they release sperm into the water column, there's, diffusion, dispersal, all sorts of things that happen. So It's definitely that are also optimization success in a lot of temperature effects. I don't have graphs to show you of these, but we do know from work that's been done over the last couple of decades that if the temperature is high enough that it's towards the thermal limit, you can get embryonic aberrations, you can get reduced larval motility, you can get reduced larval survivorship and settlement, reduced precompetency periods, but again, responses are varied. There's some species where you see effects, there's where you see positive effects, so it's really important to understand your system, your species, what the current conditions are, how close they are to their physiological limit, et cetera, to really contextualize your system. Okay, so moving on to settlement ecology. Again, recognizing that this is a sophisticated interaction that happens between the larva and the substrate. So we want to both look at whether it's impacting the physiology of the larva and its ability to actually undergo metamorphosis, but also its interaction with the substrate, because they're very actively searching around for certain cues, either on the CCA, the microbial community, etc. So we did, to look at the physiology of the larva, we did respiration experiments where Chris Langdon at the University of Miami had these tiny little respiration chambers that we would put coral larvae in and then monitor oxygen evolution and try to understand whether carbon dioxide stressed the metabolic performance of these larvae. And we found across the board that it did. So these larvae, there was metabolic suppression in the larvae when they were exposed to acidification. And this is a common thing physiologically that's been shown to occur in squid and crabs and a variety of different marine invertebrates, metabolic suppression and exposure to acidification. But what we wanted to know is, does that actually translate into an effect on fitness? Is it compromising their ability to settle and grow downstream? So we took these larvae and we conducted settlement experiments, and what we found was that as long as the substrate quality was intact, they were still able to settle fine. So there's metabolic suppression, but they're still able to undergo settlement. They're still able to metamorphose. But what was concerning is that if you remember at the beginning of the talk, crustose coralline algae, which we all know is a common settlement cue for a lot of different marine invertebrates, is high magnesium calcite, which is a more soluble form of calcium carbonate. So it's even more sensitive to ocean acidification. And so what we found was when we would condition settlement tiles under acidified scenarios, you wouldn't get the coverage of CCA that you would under ambient conditions. So in the bottom left here, we have two settlement tiles that were conditioned under ambient conditions, and then elevate. You can just visually see much coralline algae there. And what we found was when we would offer these tiles to the larvae, they just simply weren't able to find the cues they needed in order to settle. So it's this interaction between the actual substrate quality and the larval condition that is reducing or compromising larval settlement. So we had about a 70% decrease in the coverage of crustose coralline algae under acidified conditions that would translate into a 40 to 60% reduction in settlement. And there's some researchers in Australia that have continued to work with Pacific species and found I believe this was Acroporus alago, and they found when they offered them 3 different species of crustose coralline algae, or genera, I guess, species of crustose coralline algae, they found across the board with all different species that there was reduced settlement at increased CO2. So it's interfering with this interaction between the larva and the substrate. So if they do settle, how do they grow? We already talked about how acidification compromises calcification. And like Dirk was saying in his talk, we can kind of help shepherd them through these early life history stages, but at some point you have to outplant them and they have to be able to grow on their own. And that's one of the most critical bottlenecks, is their ability to grow fast enough to outcompete algae, to outcompete herbivory, all these other stressors that are on the reef. So these are Parietes asteroides larvae that we settled. They're both 3 days old, and then they were reared for 48 days under different CO2 conditions. And on the top right is an individual that was reared at ambient CO2 And you can see it's a really healthy individual. It's undergone budding, and it has about 4 or 5 new mouths there. And in the bottom right is an individual that was kept at high CO2. And so the visual difference is pretty striking. And there was also, when you actually quantify the difference in lateral extension or calcification across a variety of different Caribbean species, so Parietes asteroides, Favia fragum, and Agaricea agaricides, we saw consistently reduced growth rates with increased ocean acidification. Again, colleagues in Australia, I'll show this first. So if you take these and you look at a finer scale, like ultrastructurally, what's happening at a crystal level with these, so these are electron microscopy, scanning electron microscopy slides. Same two individuals, ambient and high CO2. And if you zoom in, these are tips that were purposefully broken off. On the left, with the ambient CO2, you can see surrounding that tip is a very smooth surface. And on the right, it's pretty rough and pitted. And if you zoom in, you have long acicular needles. Those are individual aragonite needles that have been secreted. And then what should happen is that they kind of are secreted in a very organized fashion and in a way that they can coalesce together and form a very structurally sound skeleton. On the right, we found that in elevated CO2, the actual morphology of the crystals would change. So it became, instead of long and acicular, it was more rhomboid and chunkier, and that what we think was going on was that they weren't forming in an orientation or an organized pattern enough that they could actually coalesce and form this solid structure. And so that begs the question, is it actually compromising the structural integrity of these individuals? And then in an era where we have increased storm intensity, frequency, what does that mean for their ability to withstand storm surge and other physical stressors? Okay, so I couldn't get these movies to play in these are a couple of movies. What do we have to do here? Of individuals. So these are coral recruits of Acropora specifera from Ecology in Australia that grew these corals for a month under ambient conditions and then imaged them with high resolution microscopy. And this is a really healthy, beautiful little coral spat that was grown out for the period of 30 days. And then I'm going to show you one that was reared under elevated CO2. That's 30 days with just CO2, not even adding temperature into the mix at this point. So pretty striking differences in their ability to grow. And so again, Australian colleagues looked at whether this compromised ability to grow translated into an effect on survivorship. What they found is that basically prolonging the amount of time that this little coral spat has to be in this really vulnerable stage, that it's increasingly susceptible to herbivory and all these other acute stressors. So they did outplanting experiments where we looked at herbivory effects on these recruits and found in the middle, we have fish, so bunny sturgeon fields were particularly striking, but they basically found that because they are spending longer time in a smaller state, that they're more susceptible to predation and which is necessarily surprising. And they found that compared to ambient on their side, in order to escape grazing mortality. Coral, and look at across the board what the impacts were. We saw a 12-13% reduction in fertilization success in growth. So remember, these are cumulative effects. So we're looking at we compared to ambient conditions, and then only 55% of those guys are going to actually settle. That means only 48% compared to normal conditions of individuals are even going to be able to make it to the reef and metamorphose. And then we saw the additional post-settlement impacts of growth and survivorship. So these are pretty concerning that acidification and warming exacerbate these existing bottlenecks. So, pretty depressing. What can we do? I think there's a lot of research for stressor experiments to understand. There's a lot of things that haven't been included, basically, in the experiment. What we're trying to look at now is more multi-stressor experiments to see how interactions between factors. Again, temperature will sometimes mitigate CO2 effects. So when does that happen? A lot of times, so far, there's been 1 to 2 studies that show that for subtropical species that are not already at their thermal maximum, they can withstand more temperature increases, and that there might be a little bit of an effect. But some more tropical or lower latitude species that might be at their thermal limit already, that maybe that mitigation effect isn't there. So it depends on what your existing and prevailing environmental conditions are in terms of what your response is. Another area of hope is that everyone's looking into right now is really understanding, again, genotypic and species-specific effects. And Madeline's going to speak to this later today, but I mean, we've all seen, you'll go out onto a reef after a bleaching event and you'll see 2 corals right next to one another. One bleaches, one doesn't. And so what is it about that at a genetics level, an epigenetics level, what is it about that one individual that infers resistance? And so trying to understand that, and then can we harness that knowledge and use it in our restoration efforts to try to shepherd corals through these? So I think we're doing the things we need to do to be able to address these from a research side of things and from a restoration side of things. We need to make sure we're feeding the science into the restoration practices. So for the stages we can help with, we need to help shepherd corals through. So fertilization success. If things don't seem to matter, like temperature or CO2, when there's enough sperm there, then through workshops like this, like C-Core, we're helping them through that process. We're taking them out, we're giving them the optimal sperm concentrations, we're getting the best fertilization success we can, and then we're kind of jump starting that phase. So trying to jump start them as much as we can is, I think, really critical. And then I would like to advocate that where possible, we start increasing our environmental monitoring with our ecological monitoring because of these concepts that prevailing environmental conditions really matter. That if you, these studies that show, from Emily Howells had a study in 2016 that showed that individuals that were in areas with really large temperature fluctuations were less fecund. So trying to understand your particular system, putting out temperature loggers, I'm advocating for pH loggers, and again, those are not super affordable right now, but we had the X Prize last year that was supposed to incentivize groups to develop a more economical and reliable pH sensor. So hopefully that will be coming online sometime over the next year, that people could regularly implement into their monitoring protocols. And then when outplanting, just considering that information as well. Considering how much does your temperature fluctuate in that area? How much is your CO2 fluctuating in that area? And taking that data as a piece to the puzzle in terms of where you choose your sites. And then also just continued focus. This is something that, preaching to the choir here, but on mitigating local stressors like overfishing, water quality, things that we really, because cutting carbon emissions is something that seems so not tangible to us at a local scale. And so we have all these different stressors, and the ones that we can tackle, we should, so that we give these organisms the best possible chance and ease the pressure on the system as a whole.